Welcome to the deep dive. Today, we're strapping in for a journey into the body's ultimate frontier, the human gut. This isn't just about, digestion or the simple transit of food. This is a complex, chemically volatile environment, a true war zone.
It really is a war zone where trillions of essential microbial cells are constantly fighting to maintain this delicate equilibrium against external threats.
Ironically, against the very medicine we use to save ourselves.
That's a perfect way to frame it. Our mission today is to pull apart two monumental threads of recent research that really define this battleground.
Okay.
The first is this breathtaking, sophisticated search for the surgical strike antibiotic. A drug designed with molecular precision to eliminate only the pathogens.
While leaving the good guys, the beneficial microbial community, completely untouched.
Exactly.
And the second thread.
It's the reckoning. It's facing the profound, often long-lasting collateral damage caused by the traditional broad-spectrum drugs we rely on every single day.
And as we're going to see, these drugs don't just cause resistance. They fundamentally dismantle the chemistry of the gut, rewriting the rules of metabolism and even shaping our own immune defenses from the ground up.
And the sources you've gathered are just a phenomenal synthesis of these cutting-edge fields. We're looking at molecular design, like the structure of a novel drug named lolamazine. We're looking at large-scale human genomic studies tracking the terrifying speed of resistance evolution in real time. Detailed metabolomics showing these catastrophic chemical shifts. And then high-level immunology demonstrating how simple bacterial metabolites like short-chain fatty acids and modified bile acids are direct signaling molecules to our own immune and metabolic systems.
So let's start with that central conflict, the paradox that really hangs over modern medicine. Broad-spectrum antibiotics are medical miracles. They're essential for treating systemic infections.
Absolutely.
But in the gut, they are agents of destruction, creating two distinct escalating crises.
It's exactly right. The first crisis is the acceleration of antimicrobial resistance, or AMR. By bathing the entire gut community in a non-specific lethal dose, we're applying the ultimate selective pressure.
You're just ensuring that any surviving resistance gene is amplified and persists.
And the second is the immediate destruction of what we call colonization resistance.
Yeah.
You wipe out the good bacteria, and you instantly become highly susceptible to opportunistic secondary infections.
And the most notorious of which is C. difficile infection, or CDI.
That's the one. So today we're going to dissect recent breakthroughs that finally offer a scientifically guided path out of this dilemma, starting with that dream of precision.
Okay, let's unpack this concept of the precision antibiotic. What is the fundamental problem that researchers are trying to solve when they look at drugs like, say, ciprofloxacin or amoxicillin?
Problem is one of massive overkill. It really is, particularly within the world of gram-negative bacteria.
Okay.
When you take a traditional broad-spectrum antibiotic, it's like using a nuclear bomb to take out a single sniper.
Right.
It kills the target gram-negative pathogen. Maybe it's a specific strain of E. coli causing a UTI. But it also indiscriminately wipes out the entire population of beneficial gram-negative commensals in your gut.
And why are those specific gram-negatives so critical? What are they doing for us?
They provide colonization resistance. These bacteria, often species within the bacteroids or specific enterobacteria groups, are occupying the physical niches.
Taking up space.
Yes, taking up space and consuming the exact nutritional resources that would block an invader like C. difficile. So when you remove them, you leave a vacuum.
You leave an opening.
The huge opening. The goal, therefore, has to be finding a molecule that can somehow distinguish between the bad guys and the essential good guys, specifically within that gram-negative world. And this is the whole rationale behind the development of lolamazine.
The name lolamazine sounds almost whimsical, but the science behind it is anything but. This search for selectivity led researchers to target an essential, yet often overlooked, part of the bacterial machinery, the LOLOP system.
That's right. LOL stands for lipoprotein outer membrane localization system.
Okay.
Lipoproteins are crucial components that help build and maintain the structural integrity of the bacterial cell envelope. They're synthesized in the inner membrane, and then they have to be transported across the periplasm to be inserted into the outer membrane.
So they're like the bricks for the outer wall.
A perfect analogy. And the LOL system, which involves 5 key proteins designated A through E, is responsible for this vital trafficking operation. If you stop the trafficking, the bacterium can't maintain its structure, and it dies.
Simple as that. So which specific part of that system did the researchers prioritize for inhibition? Where's the weak link?
They needed the linchpin. So initial genomic analysis revealed that LOL and LOLB were sometimes non-essential or had redundant functions.
So not a reliable target.
Not reliable enough. However, the LOL-CDE complex, which is the actual transporter machinery embedded in the inner membrane that drives this trafficking, that was identified as absolutely essential for the viability of many key gram-negative pathogens.
So that's the bullseye.
That was the molecular goal. Inhibit LOL-CDE.
Okay, let's unpack the chemistry of discovery for lilomycin, because this is where the story gets really interesting. It illustrates the biggest hurdle in developing new antibiotics. Just getting the drug across the fortified walls of a gram-negative cell.
This is the $64,000 question in modern drug discovery. They started with existing LOL-CDE inhibitor scaffolds. These compounds they labeled 1 and 2.
Right.
And they saw good activity in vitro against strains that were already compromised, specifically strains of E. coli that either lacked the efflux pump, the ADL-C strain.
Which is the pump that normally just spits the drug right back out.
Exactly. Or they tested it against strains with an increased permeability, the AFAX strain.
So if the researchers basically cheated and made the bacterial wall leaky, the compounds worked.
They worked right.
But against the wild-type, healthy gram-negative pathogens that are causing actual infections?
They were useless. Just no significant antimicrobial activity. The target engagement was fine, but the compounds simply could not accumulate inside the healthy cell. They were running straight up against what we call the gram-negative accumulation barrier.
The fortress wall.
A fortress wall. The outer membrane is a lipid bilayer protected by a lipopolysaccharide, and it's brutally effective at keeping most drugs out.
And chemical development often relies on established guidelines to cross that barrier. You mentioned the entry rules. What did those rules suggest they needed to add to make the drug accumulate?
So the entry rules define the physicochemical properties that you need for successful penetration of the gram-negative outer membrane. Successful molecules are typically small, they're rigid, they have a low number of rotatable bonds, and critically, they often require a primary amine group.
Okay, why the amine?
It helps it interact favorably with the cell surface and sort of initiate entry. And compounds 1 and 2, they lack that amine. So the logical next step was to create a hybrid scaffold and then introduce that primary amine.
And they tried it. They achieved better whole cell accumulation, right? They solved the entry problem.
They did. But here's the frustrating, often contradictory nature of organic chemistry. The addition of the amine, while it improved accumulation, proved deleterious to the drug's activity against the actual all-CDE target.
Oh, so it gets in, but it doesn't work as well.
It significantly reduced the potency needed for effective killing. They had traded one problem-poor accumulation for another poor target engagement.
They hit a wall, better entry, but worse killing. This forced them to completely rethink the conventional wisdom that high total cellular accumulation equals a good drug. And this is where the breakthrough idea of spatial targeting comes in.
This is the truly novel insight they postulated. Why do we need high total accumulation if the target isn't everywhere? The LOL-CDE complex is embedded in the inner membrane, and it acts on lipoproteins in the periplasm.
And periplasm is that tiny space between the two membranes.
The tiny aqueous space, exactly. It only comprises about 7% of the total volume of the gram-negative cell. So they thought, what if we could optimize the molecule not for massive accumulation, but for maximum efficiency within that tiny 7% compartment?
So they were optimizing for spatial delivery, not just brute force.
Precisely. They focused on structural modifications that optimize target binding and minimize toxicity, even if it meant sacrificing overall cellular accumulation. And that iterative structure-activity relationship approach that led them to lilomycin.
And the proof is in the pudding. The final molecule actually accumulated less than the failed ones.
That's the amazing part. Its whole cell accumulation in E. coli was actually below the accumulation of their failed control compounds. Yet it retained marked activity against the pathogens.
Wow.
It proves that you can bypass the traditional accumulation barriers by optimizing binding affinity within the specific target compartment.
That is a fundamental paradigm shift in how we approach gram-negative antibiotic design. So let's look at the result of that effort. Lilomycin's selective efficacy. Who does it hit?
It demonstrated potent activity, very low minimum inhibitory concentrations, or MICs, in the 1 to 8 big ml range against the heavy hitters of hospital-acquired infections.
Like what?
E. coli, Klebsiella pneumonia, and Enterobacter cloae.
Those are all major clinical gram-negative pathogens where resistance is a growing catastrophe. And the mechanism of its selectivity is built directly into its structure, targeting those specific differences in the LOL-CDE complex.
Yes. The high activity strongly correlated with the high sequence identity of the LOL-CDE complex across those susceptible species.
Yeah.
They showed 90 to 100% identity across the LOL-C, LOL-T, and LOL-E proteins. The drug fits those target locks perfectly.
So where is the precision? Who does it spare?
And this is the crucial finding. Lolomycin was almost entirely inactive, with MICs routinely exceeding 128 megaml against gram-negative gut commensals.
The good guys.
The good guys. We're talking about key protective species like Bacteroides fragilis and Faecalibacterium prosnitzi. And the reason? Their all CDE sequence similarity is dramatically lower, often ranging only from 23 to 44%.
So the drug literally doesn't recognize their machinery.
It doesn't fit the lock.
Yeah.
And crucially, it was also inactive against other non-target gram-negative pathogens like P. aeruginosa and A. baumannii,
Right.
and it spared all gram-positive bacteria entirely.
That is a molecular scalpel. It's harnessing evolutionary divergence to spare 99% of the gut ecosystem. So the final test. Does this molecular precision translate to protecting the physical environment of the gut and preventing infection?
Absolutely. The animal model data confirms that this precision maintains microbiome integrity. Mice were given lolamycin for 31 days,
Okay.
and the researchers measured bacterial diversity using metrics like the Shannon index, which reflects the richness and evenness of species.
And what did they find?
Unlike amoxicillin and clindamycin, which caused major destabilizing shifts in diversity and population structure, lolamycin caused no significant shifts whatsoever. The ecosystem just remained stable.
And the ultimate goal, preventing C. difficile infection.
Lolamycin proved highly effective in preventing C. difficile colonization and outgrowth following a direct spore challenge. The CFU counts the colony-forming units, which is a measure of how many pathogens establish themselves. And the lolamycin-treated mice were statistically indistinguishable from the healthy, untreated control group. They were protected because their colonization resistance remained intact.
Meanwhile, the mice were treated with traditional antibiotics.
Showed massive C. difficile outgrowth. This research offers a compelling roadmap for the future. Drugs designed to neutralize the threat without sabotaging the internal defense system.
We've seen the extraordinary potential of future medicine, but now we have to face the current price we are paying for our reliance on broad-spectrum antibiotics. Let's look at ciprofloxacin, a drug we use daily, and how quickly it turns our own gut into a permanent reservoir of resistance.
This specific study involving 60 human subjects tracked the impact of a standard 5-day course of ciprofloxacin, and the data revealed just an alarming speed of genetic adaptation.
How fast?
Within that short 5-day window, there was a rapid, dramatic evolution of resistance occurring not just in the pathogens, but in the innocent gut commensals.
5 days to permanently alter the genetic landscape. What was the central mechanism of this rapid evolution as revealed by analyzing thousands of microbial genomes?
The mechanism was crystal clear. Convergent evolution. Multiple, unrelated species in the gut all converged on the same solution to survive the drug.
They all found the same workaround.
The same one. They focused overwhelmingly on one gene, DNA gyrase subunit A, or GYRA.
Okay, what does that do?
DNA gyrase is the essential enzyme responsible for unwinding DNA during replication, and it happens to be the primary target of ciprofloxacin. Secondarily, resistance appeared in PARS, another topoisomerase gene.
So the pressure was so intense the mutation appeared across diverse species simultaneously. How widespread was this?
The researchers analyzed over 5,000 genomes, 5,665 to be exact, and they found that approximately 11% of the susceptible bacterial populations underwent what are called selective sweeps.
A selective sweep. That means the resistant version just takes over.
It means the resistant mutant rapidly outcompeted and replaced the non-resistant population under the pressure of the drug. The resistant strain suddenly has this massive survival advantage, allowing it to become dominant almost instantly.
Okay, a selective sweep is expected while the drug is present, but the great hope is that once the antibiotic clears the system, the resistant strains carry a fitness cost, meaning they're slightly less efficient than their wild-type cousins, and eventually they fade away. Did that happen here?
That is always the hope. But the reality was far more disturbing. The study showed that over 84% of those acquired genetic changes, those sweeping resistance mutations, remained dominant in the populations months after the antibiotic course ended.
Months later. They persist.
They persist in the majority of individuals studied.
Why? This is the critical finding about resistance persistence. Why don't they go away?
Because these specific gyro-resistance mutations that arose de novo during the study, particularly point mutations at positions, serine 83 and aspartate 87 exhibited no measurable fitness cost.
No downside.
No downside. They achieved resistance without impairing the native function of the DNA gyrase enzyme. So since there was no biological drawback, no selective pressure existed for them to revert back to the susceptible state.
So we've created a perfect, evolutionarily stable mutation.
That's it exactly. Researchers projected that it would require more than a year, possibly much, much longer, for these resistance alleles to naturally revert below a nominal frequency of just 1% in the population.
So a 5-day course of antibiotics effectively turned these individuals into long-term reservoirs of ciprofloxacin resistance.
Yes, resistant strains that could potentially transfer those genes to transient pathogens or just be amplified again during future antibiotic exposure.
And this rapid evolution is built upon pre-existing conditions. It's not just a completely blank slate, is it?
Precisely. The study found that resistance alleles at the critical gyro 0.83 position were already present in 26 of the 40 species analyzed before the ciprofloxacin course began.
Wow, so it was already there.
It was there, averaging about 34% prevalence across those species. The antibiotic merely acted as a massive amplifier. Those pre-resistant strains had elevated abundances during antibiotic exposure, demonstrating that baseline resistance determines which strains survive and subsequently thrive.
The drug just selects for the most resilient of the already present resistant strains. Let's shift from the genetic destruction to the chemical destruction, or what we called metabolomic collapse. The sources use cyphopyrazone as another example of a broad-spectrum drug. How does antibiotic use create the exact metabolic conditions necessary for a pathogen like C. difficile to take hold?
The drugs don't just kill, they dismantle the carefully balanced chemical environment that provides colonization resistance. The gut is shifted into this susceptible state known as S1, characterized by massive, system-wide metabolic disruption that clears the way for the invader.
The bile acid flip sounds particularly critical for C. difficile viability.
It is the ignition switch. C. difficile spores require specific signals for germination. Healthy commensals typically take primary bile acids synthesized by the host liver-like terocolate and modify them into secondary bile acids like deoxylate.
Right.
The susceptible S1 state showed an increase in primary bile acids, especially terocolate, which is a potent C. difficile germination trigger.
So the trigger goes up.
And at the same time, the commensal community that performs the protective modification of these bile acids has been wiped out, leading to a simultaneous decrease in the protective secondary bile acids.
So the environment is chemically set up for the pathogen to thrive.
Absolutely. The gut essentially becomes a feast. The beneficial microbes that were eliminated were largely responsible for carbohydrate fermentation. When they die, the substrates they would have consumed surge dramatically.
How dramatic.
Carbohydrates and sugar alcohols just spiked. Mannitol increased 553-fold, and sorbitol increased a truly astonishing 1,053-fold.
Wait, 1,000-fold increase in sorbitol? That's not a subtle chemical shift. That is the equivalent of turning up the lights and serving a multi-course, all-you-can-eat buffet for the pathogen.
That's a great way to put it. Sorbitol is a simple sugar alcohol. In a healthy gut, beneficial bacteria rapidly consume and metabolize these things. But C. difficile has the metabolic machinery to utilize these molecules when its competitors are gone. This massive surge is the direct result of losing that competitive beneficial community.
And the loss of the beneficial community is confirmed by the dramatic drop in their primary metabolic output.
Precisely. This loss of fermentation was confirmed by a huge 66-fold decrease in crucial short-chain fatty acids, or SCFAs, like valerate. Low SCFAs mean lost colonization resistance, lost energy for the host colonocytes, and lost immunomodulatory signals.
Right on top of that.
On top of that, you have a surplus of amino acids essential for C. difficile growth, glycine, proline, tryptophan. The environment is just metabolically perfect for spore germination and outgrowth.
And they confirm this in the lab.
They did. Ex vivo experiments validated this. C. difficile spores germinated readily in the scaecal contents harvested from antibiotic-treated mice, but were suppressed in contents from healthy controls.
Now, this danger isn't limited to antimicrobials. Research suggests that non-antibiotic drugs, which you might take for heart issues or depression, can also compromise colonization resistance.
Yes, and this is a really sobering extension of the problem, because we tend to quarantine our thinking about resistance to just antibiotics. Researchers tested 53 commonly used non-antibiotic drugs on synthetic microbial communities.
These COM20 communities.
Right. And they found that 15 of them, including some widely used antidepressants and heart medications, promoted the expansion of the pathogen S. typhimurium.
That sounds completely unrelated to their intended function. What was the unifying factor that allowed these non-antibiotic drugs to open the door for infection?
The unifying factor was shockingly simple: total community biomass. The researchers found that community biomass, measured by optical density, was intensely and strongly negatively correlated with pathogen growth. The correlation coefficient was Spearman's art 0.98.
Minus 0.98. That level of correlation it's nearly perfect.
It's almost a straight line.
So if a drug, regardless of its primary target, reduces the sheer density and population size of the beneficial residents sufficiently, it compromises the structural integrity of the ecosystem.
That's the takeaway. It highlights a critical vulnerability. Colonization resistance is not just dependent on specific antagonistic chemicals. It's a numbers game. Any substance that sufficiently reduces the total microbial load allows the metabolic disruption to occur, opening the door for infection.
So the bile acid flip, or the sugar surge we saw with cefoprazone, could potentially be triggered by common non-antibiotic drugs if they just impact microbial density enough.
That's what the data suggests.
We've established that the microbiome is a chemical factory influencing colonization resistance and host metabolism. Now, let's pivot to its role as an active schoolhouse for our immune system, specifically focusing on how it dictates the behavior of key T-cell populations that maintain tolerance.
This is where we move beyond simple defense and into instruction, and we're focusing on two critical interconnected T-cell populations: regulatory T-cells, or Tregs.
The peacekeepers.
The peacekeepers, exactly. They actively suppress inflammation and enforce immune tolerance, preventing autoimmunity. And then T helper 17 cells, or Th17s, which are pro-inflammatory first responders essential for mucosal defense against extracellular pathogens.
And the microbiota instructs the maturation and localization of both.
Both of them, yes.
Let's start with the Tregs, the peacekeepers. What is the direct molecular signal that the microbes use to promote Treg generation in the colon?
The key signals are the short-chain fatty acids, the SCFA, so acetate, propionate, and most famously, butyrate. These are the result of beneficial bacterial fermentation of dietary fiber. And the evidence is just crystal clear. SCFAs are essential for promoting the generation of peripheral Tregs,
Particularly the ones in the gut lining.
especially the FOXP3 plus Treg populations found in the colonic lamina propria, the tissue layer right beneath the epithelium.
So what is the mechanism? How does a simple fatty acid tell an immune cell what to do?
Well, SCFAs are small enough to be absorbed by the colonocytes and enter the immune cells. Once they're inside, they act as metabolic regulators, specifically affecting histone deacetylases, or HDACs.
Okay.
Butyrate, for instance, inhibits HDACs, which leads to hyperacetylation of histones, essentially relaxing the chromatin structure around the FOXP3 gene.
So it makes the gene easier to read.
It makes it accessible. This epigenetic modification promotes the expression of the FOXP3 transcription factor, which is the master regulator for the Treg lineage. So the bacteria are using their metabolic byproducts to literally epigenetically program our T cells toward tolerance.
That is just stunning. And if you strip away the bacteria and those SCFAs with antibiotics, you can restore that tolerance simply by putting the metabolite back in.
The research confirmed this strong causal link. When butyrate was administered to antibiotic-treated mice, which had lost their natural Treg populations, it successfully restored the FOXP3 plus Treg populations in the colonic lamina propria. This confirmed that the bacterial output, the SCFAs, are not just correlated with tolerance. They are a necessary, sufficient, and direct molecular signal for local immune regulation.
Okay, now let's look at the TH7. They're often characterized as driving inflammation, and they are surprisingly abundant in the small intestinal lamina propria, or SILP. What controls their presence?
Their accumulation is entirely dictated by the presence of the microbiota. Germ-free mice completely lack T17 cells in the SILP. Completely gone. This led researchers to hunt for the specific bacterial group acting as the master inducer, and the discovery hinged on one specific structurally unique group: segmented filamentous bacteria, or SFB.
SFB. These bacteria are famous for their incredibly tight physical attachment to the gut wall, almost like Velcro. How definitive was the link between SFB and Th17 induction?
It was proven through single-species colonization experiments. Researchers took germ-free mice and colonized them solely with SFB. This single-species colonization was sufficient to induce a robust accumulation of Th17 cells in the intestinal lamina propria.
Just that one bug.
Just that one. And crucially, this effect was highly species-specific. Colonization with other common bacteria like Bacteroid species or various Clostridium mixes did not induce Th17 cells.
So, SFB are the architects of adaptive immune readiness in the small intestine, forcing the host to develop these necessary defense cells.
That's a great way to put it. The colonization induced a powerful immune response program in the terminal ileum, including the activation of genes directly associated with Th17. SFB provide an essential localized danger signal or presence signal, ensuring the SILP is populated with 17 cells.
Which are crucial for fighting certain bacterial and fungal infections. But Th17 cells are pro-inflammatory. They're essential for defense, but they're potentially destructive. If the microbiota is forcing their existence, how does the body prevent constant low-grade gut inflammation? This is the remarkable part. The small intestine seems to tame the T cells after they are induced.
This functional shift is one of the most exciting aspects of mucosal immunology. The small intestine environment post-SFB induction is where these pro-inflammatory Th17 cells are actively tamed. They acquire a suppressive phenotype and are therefore referred to as RTH17 cells, regulatory Th17 cells.
Okay, so what are the specific functional details that define this transition from inflammatory to suppressive? How do they change?
These tamed RTH17 cells, they retain their initial Th17 lineage markers, like RORγt and IL-17A, so they still look like a Th17 cell, but their functional chemical profile changes drastically. They exhibit greatly reduced expression of the strong pro-inflammatory cytokines, specifically TNF-E and IL-2, and most importantly, they simultaneously ramp up expression of the crucial anti-inflammatory cytokine IL-10. They flip their function. This functional change allows them to suppress the proliferation of other T cells in vitro, essentially acting as peacekeepers derived from a potentially inflammatory line.
That demonstrates localized education at its finest. The environment dictates function, not just lineage.
And we have definitive proof that the small intestine environment itself is essential for this taming process. If researchers prevented T cells from migrating to the small intestine, for example, by studying mice deficient in the CCR6 gene,
Which prevents migration to the SILP.
right, those T cells failed to develop the suppressive phenotype. They remained purely pro-inflammatory. In fact, when transferred into a host, those untamed cells could cause severe inflammatory bowel disease.
So it's proof positive.
It confirms the small intestine is the critical classroom for acquiring regulatory tolerance from these necessary but potentially dangerous first responder cells.
We've seen the power of chemical signals like SCFAs. Let's pivot back to bile acids, but look at them not just as infectious triggers, but as microbially controlled host signals that influence systemic metabolism. And to truly understand this, researchers first had to overcome the limitation of traditional stool sampling.
Right. Stool samples are fundamentally misleading for studying dynamic processes. They only provide a cumulative homogenized view of the final output from the end of the colon.
It's the end of the story.
It's the end of the story. The key chemical dialogue between the host and the microbes, the deconjugation and modification, happens dynamically as the bile acids travel through 30+ feet of intestine. So to get true regional profiles, researchers developed an ingenious tool: passive sampling capsules.
These capsules sound incredibly sophisticated. They're designed to open based on pH changes along the intestinal tract.
Exactly. Different devices were coated with enteric materials that dissolved at progressively higher pH levels. This allowed researchers to capture small regional fluid samples up to 400 microliters from the duodenum, the jejunum, the ileum, and the ascending colon in vivo during the natural process of digestion.
Which gave them an unprecedented dynamic chemical map of the gut.
That's right.
So what did these regional samples immediately reveal about how bile acids are processed compared to the static view we had from stool?
Well, they mapped the gradient of processing. The first major finding was the significant drop in total concentration of bile acids as they moved down the tract. But more importantly, the samples mapped the active zones of microbial transformation. The percentage of liver-conjugated bile acids, like TGF-β, TCA, and glycolic acid, GCA, which are the host's primary output, decreased progressively as the samples moved down the gut. This demonstrated that microbial deconjugation activity was occurring continually, not just starting en masse in the colon.
And the typical view derived from stool, that secondary bile acids dominate everything, was fundamentally incorrect for most of the small intestine.
Completely misleading. Secondary bile acids like deoxycholine-DCA and lithocolic acid LCA dominate stool samples, which led to the assumption that they dominated the entire lower gut. But the regional intestinal samples showed they did not dominate the upper regions, confirming that the intensive microbial transformations leading to those secondary bile acids happen progressively throughout the tract.
Stool only gives you the final product, which obscures the dynamic nature of the chemical factory.
Precisely.
Furthermore, these devices allowed the researchers to correlate the concentration of specific bile acids with the presence of specific bacteria, pinpointing the active microbial players in that initial deconjugation phase.
That's right. This level of granularity allowed them to make causal links. Specific bacteria, including Elastipes putridinus and Balafla wadsworthia, showed strong negative correlations with the presence of primary conjugated bile acids in the intestinal samples.
Which is crucial.
It's crucial. It confirms these species are major players in breaking down the host-produced bile acids early on, often far before the contents hit the terminal colon. For example, the decline in TCA concentration along the tract correlated negatively with the abundance of those specific Elastipes and Balafla species in the device samples. It offers a much clearer picture of their functional niche than stool data ever could.
Beyond mapping the known transformations, this intensive chemical scrutiny led to the discovery of entirely new chemistry. The sources mention a fifth mechanism of microbial bile acid modification, something the host never produces.
This was a monumental finding. For decades, we had cataloged 4 primary ways microbes modify bile acids: dehydroxylation, dehydration, epimerization, and deconjugation.
Which is removing the taurine or glycine.
Right. But researchers discovered a novel, fifth mechanism, the amide conjugation of the chocolate backbone with other amino acids, specifically phenylalanine, tyrosine, and leucine.
So the bacteria are stripping off the host's conjugated tags and retagging the bile acid backbone with a completely new chemical entity derived from the diet. That sounds like true chemical mischief.
It is elegant, complex chemistry. Specific Clostridium bolti strains were shown to synthesize these novel conjugates, fenchal and tericol, both in culture and critically in vivo.
Had they proved that?
They verified this by feeding mice labeled 13C phenylalanine and then detecting the labeled fechol in their feces. This proves that they are taking dietary or endogenous amino acids and using them as precursors to create a novel host signaling molecule.
So these are novel, microbially synthesized molecules the host body has never encountered through its own metabolic pathways. What is the impact of these new derivatives on the host system?
They are powerful modulators of host receptors. They focused on the farnesoid X receptor, or FXR.
FXR.
FXR is a crucial nuclear receptor found in the intestine and the liver. It acts as a master transcriptional switch for controlling lipid, glucose, and cholesterol homeostasis. Normally, native bile acids activated to regulate the entire body's energy balance.
The gut bacteria are creating super stimulators that hijack this master switch.
Exactly. These novel microbially conjugated bile acids, fechol and tericol, turned out to be potent FXR agonists. They're designed to fit the host's lock better than the host's own key.
How potent are we talking?
Astonishingly potent. The tyrosine conjugate, tericol, was identified as the most potent agonist identified, with an EC50 of 0.14 micromolar. It was significantly stronger than the native ligand, chenodeoxycholic acid.
So the host's own bile acids are weak signals compared to these microbially generated compounds.
Much weaker.
And this wasn't just in vitro novelty. This has systemic impact in vivo.
When researchers administered these novel conjugates to mice, it led to a time-dependent, dose-dependent reduction in the expression of key liver bile acid synthesis genes, specifically CYP7A1 and CP8B1. The classic negative feedback loop mediated by FXR activation, the microbial chemistry derived from dietary components and mediated by Clostridium bolti, is directly signaling to the host's liver and telling it to stop synthesizing bile acids. It's definitive proof that microbial chemical output is not just waste or fuel. It is actively controlling fundamental host metabolic regulatory pathways. Hashtag outro up.
This deep dive has woven together an incredible tapestry of modern microbiology, chemistry, and immunology. We began with the promise of the future, embodied by surgical strike antibiotics like lalamicin, which demonstrate that we can exploit minute evolutionary differences in essential bacterial machinery, like the LOL-CDE complex, to spare the integrity and diversity of the gut microbiome.
And ensure robust colonization resistance against pathogens like C. difficile.
And then we confronted the immediate, alarming price we pay for nonspecific medicine. We saw how ciprofloxacin drives the rapid, fitness, cost-free evolution of resistance, leading to mutations in gyre that persist for months, making us permanent carriers of resistance.
And we demonstrated how broad-spectrum drugs cause a profound metabolic collapse, flipping the bile acid switch to trigger infection and serving up massive surpluses of sugar alcohols, a feast that non-antibiotic drugs can also create simply by reducing total microbial biomass below a critical threshold.
And finally, we mapped the complex immune schoolhouse of the gut. We learned that the short-chain fatty acids are essential signals for tolerance, directly restoring crucial FOXP3 plus Treg populations by epigenetically modifying host DNA.
While at the same time, specific groups like segmented filamentous bacteria educate RT817 cells, forcing them to mature into suppressive RT817 cells in the small intestine, thus teaching the immune system the difference between defense and self-attack.
The overarching lesson here is so clear. The integrity of your microbiome is paramount. It is not just about digestion, it is a dynamic chemical factory that produces essential signaling molecules that regulate our immunity, our metabolism, and our resistance to infection.
And these signals, be the SCFAs influencing T cells or novel amino acid bile acid conjugates controlling master host switches like FXR, they're foundational to long-term systemic health.
We've established that gut microbes, using only simple dietary precursors, can synthesize bile acid derivatives potent enough to strongly modulate critical host receptors, like the FXR, overriding the host's own regulatory signals. So here is the provocative thought we leave you with. If gut microbes can so easily and profoundly hijack our fundamental metabolic pathways using novel chemistry, imagine the vast landscape of uncharacterized microbial metabolites that might be secretly controlling our endocrine and neurological systems right now. What primary host regulatory pathways, which we currently think are exclusively host-controlled, perhaps appetite hormones, sleep cycles, or mood regulation, might actually be under daily, profound, and unseen influence by the trillions of guests inside us? The internal frontier is far more complex than we ever imagined.